The neuron is the core of human control. It transmits high-speed bioelectrical signals. Pathological decay triggers degeneration.
The neuron is characterized by distinct morphologies. These structures dictate their functional roles. Neurons are classified by their polarity, from bipolar to multipolar.
The soma serves as the manufacturing center. It contains the nucleus and genetic material. Neurons are characterized by an extensive rough endoplasmic reticulum.
The Golgi apparatus acts as a cellular refinery, while mitochondria generate vital ATP.
The neuron depends on a system of filaments. Microtubules act as tracks for internal transport. Neurofilaments provide crucial mechanical strength, and microfilaments remodel the membrane.
Neurons have one axon and multiple dendrites. Bipolar neurons feature two distinct processes. Unipolar cells have a single branching process. Pseudo-unipolar neurons are primary sensory cells. The mushroom spine acts as a compartment, isolating signals received at one synapse. The journey begins with proliferation and ends with successful integration into a functional circuit. Dendrites are the primary receptive structures. They branch to increase surface area. Dendritic spines are small protrusions where synapses are located. These neurogenesis generates new neurons from stem cells. In niches like the SVZ, they undergo asymmetric division. This signal thrive or undergo apoptosis.
Once born, a neuroblast must travel to its final destination. Guided by glial fibers and chemical errors create heterotopias, potentially leading to epilepsy. The neuron undergoes differentiation. It extends its axon toward target cells. Myelination then refines the transmission, enabling fast saltatory conduction. Filopodia are long and thin, serving as precursors to stable synapses.
Dendritic spines are crucial for brain function. Thin spines have a long neck and a small head. They are learning spines capable of strengthening. This plasticity is the foundation of memory.
Stubby spines lack a traditional neck. The head attaches directly to the dendrite. They provide stable, high-conductance pathways. They are common in early development. Dendritic spines come in various shapes. Mushroom spines have a large head and a narrow neck. They are stable and store long-term memories. Neurons coordinate functions through electrical excitability. At rest, a neuron maintains and actively moves ions to maintain this state. Potasks out, leaving the interior negative.
The potassium channels close slowly. This causes the potential to become more negative than the resting level briefly. This prevents another propagates in only one direction. When a neuron receives a stimulus, the membrane potential changes. Na+ channels open rapidly, allowing a massive influx of ions. This triggers an action potential. When an action potential reaches the terminal, it triggers the opening of voltage-gated calcium channels. This causes vesicles to release neurotransmin to the synaptic cleft to bind with receptors.
Sensory receptors collect data. This is afferent transmission. Interneurons integrate the signal, sending commands back via efferent nerves, resulting in an immediate reflex action, while simultaneously informing the brain.
Meet acetylcholine, a powerful chemical messenger. It is usually classified as excitatory. It acts at the neuromuscular junction, where it directly triggers muscle contraction. Glutamate is an excitatory neurotransmitter. It is critical for learning and memory.
Meet GABA, your brain's primary inhibitory neurotransmitter. Its main function is preventing neural overexcitation. It stops chaos and maintains a state of calm.
Neurotransmitters. Today we look at dopamine. It is classified as modulatory. It is central to brain reward pathways and controls motor function.
We can record the body's electrical activity. An ECG records the heart's activity. Standard leads form Einthoven's triangle. These include ECG and EMG, providing a window into health. To gain more views, 3 augmented leads are used. AVR connects to the AVF, complete the frontal plane. This increases the signal's voltage by 50%, making waves easier to analyze. A normal heartbeat produces a characteristic wave pattern. The P wave represents atrial depolarization, the QRS complex ventricles. The T wave represents repolarization of the ventricles.
The EEG records the summation of postsynaptic potentials. These signals are analyzed by their frequency bands. They correlate with different states of consciousness. EMG measures muscle electrical signals. ERG assesses the retina's response to light. EOG evaluates eye movements.
The body generates weak magnetic fields. Specialized squid sensors measure them.
MEG offers high-resolution brain maps. MCG provides a non-contact heart scan.
The body generates magnetic fields. They are exceptionally weak. MEG maps brain activity, while MCG measures the heart, offering non-contact alternatives.
Neuronal death is a normal part of development. Pruning eliminates weak connections to wire the adult brain. But disease causes premature and unnatural decay. Understanding Parkinson's disease. It involves the death of dopamine neurons in the basal ganglia. This leads to tremors and slow movement, as well as severe balance issues.
Huntington's disease is a genetic disorder. Glutamate overproduction causes excitotoxicity. This leads to uncontrollable movements and cognitive decline.
Alzheimer's disease destroys the brain. Amyloid plaques and tau tangles accumulate, blocking cell communication. This triggers memory loss. It destroys the capacity for everyday life. Spinal cord injury can sever axons, causing permanent paralysis. Dying neurons release glutamate, triggering a wave of death in healthy cells.
Neurology is becoming regenerative. Researchers are manipulating stem cells to replace lost neurons, aiming to stimulate the brain to heal from within, creating specific neurons for Parkinson's patients.
Brain neurogenesis is not fully understood. While new neurons are born in the hippocampus, stress and aging can inhibit this growth, but exercise and enriched environments enhance it, aiding cognitive longevity.
By recording neural signals, researchers can now decode the pure intention to move, effectively bypassing damaged pathways. From the microscopic architecture of a dendritic spine, when this balance refound, but the same plasticity provides the foundation for cures.